Non-invasive imaging of both brain structure and activity is amongst the most important technological advances in neuroscience research. It has allowed us to ask questions about the living human brain that were not possible 50 years ago. But good data from neuroimaging depends upon solid research design, so we'll explore some issues that arise with this type of research. There are many techniques to assess brain structure, but we'll focus on those that measure brain activity. fMRI is powerful because it gives detailed images of brain structure as well as measurement of oxygenated blood flow, taken as an indication of neuronal activity. fNIRS operates on a similar concept, except it detects blood flow by measuring how it responds to infrared light shown from the scalp. PET uses a radioactive tracer to measure metabolic or biochemical activity. If oxygen is radioactively tagged and injected, it can also be used to track oxygenated blood flow in the brain, but it doesn't produce the detailed images like fMRI. PET can also be done using radioactive glucose to assess neuronal energy use. EEG measures electrical activity more directly from electrodes placed on the scalp, so is technically not a neuroimaging method because it doesn't produce pictures of the brain. However, it's excellent for assessing changes on a fast timescale. The power of neuroimaging is highlighted by this 2006 study of a patient in a vegetative state. Using functional MRI, researchers found that when asked to imagine certain actions, such as playing tennis or walking around her home, she showed the same brain activation as controls, without head injury, shown for comparison on the bottom. The fact that she could respond with her own brain activity in an intentional, purposeful way allowed her to communicate despite her inability to move. But in order to do these miraculous things with neuroimaging, there are some important research design considerations. One problem with neuroimaging research is the amount of noise in the data. The brain is always busy, so the activity you're interested in is buried under all sorts of other thinking. To deal with this, researchers usually do a number of trials for the tasks participants are asked to do. That way, if there's a consistent response across trials, it should show up every time within the noise. Another important component of imaging is subtracting the noisy thinking from the activity of interest, so results are almost always displayed as a comparison to some control condition. This subtraction of baseline control activity usually requires within subjects' measurements, which is helpful because individuals' brains vary a lot. For instance, the location and size of the bumps and grooves on the cortex. In making between subjects comparisons, researchers often use similar participants who are matched on several key characteristics like age, gender, and handedness. We'll consider how these problems and solutions impact research design. A fantastic demonstration of noise in brain activity is shown by EEG. Baseline EEG looks like squiggles of spaghetti. This here shows electrical changes in millivolts over time in one of the electrodes placed on my head. Notice in the photo that there are a whole bunch of electrodes, and each of these produce an activity trace shown in the panel on the right. Notice the deflections and dark areas that show up at certain points. These were produced by blinks, and this happened when I clenched my teeth. All sorts of these types of events are happening in the brain, so EEG data is messy. However, if someone carries out a task many times and the EEG traces are added together, it's possible to see the signals in the noise. These are called event-related potentials, or ERPs, and they signify brain reactions to particular stimuli or events. Let's look at one of these ERPs. This shows electrical activity picked up by EEG in a semantic violation task. The red is during a control condition when the participant hears a semantically correct sentence, like, "I swept the floor with a broom." The blue is during a sentence that doesn't make sense. "I swept the floor with a baboon." This is called semantic violation. You can see these lines overlap, But at around 400 milliseconds after the last word was heard, there's a noticeable difference. To see what this semantic violation looks like, the waveforms are combined so that the noise is subtracted out. What's left is the brain's activity specific to that task, called a difference waveform. By averaging the difference waveforms from many individuals, you see fewer squiggly lines, and there's a large negative electrical response at around 400 milliseconds, called the N400. This task and technique have been used in coma patients to assess consciousness. If someone's brain responds to sentence meaning in this way, it suggests that they're conscious. One of the most important parts of any experiment is choosing the appropriate controls, which can be tricky with neuroimaging research. In this study, researchers were interested in brain activity associated with the rewarding properties of music. So they recruited some musicians and scanned their brains while listening to music they loved. In fact, they were asked to choose a piece of music that reliably sent chills up their spine. So what can be used as a control condition? Silence? If there was a difference in brain activity, it would be difficult to determine whether it was due to just hearing music or specifically the music that moves you deeply. So the researchers did a clever thing to get a sensible comparison. For the control condition, each participant heard the chills up your spine music from another participant, which meant any differences in the dependent variables could not be due to just hearing music. To ensure that this control worked as intended, they measured heart rate, shown in the top panel, and EMG, which measures muscle tension on the bottom. The two bar graphs on the left show that both heart rate and EMG were higher on average for participants while listening to their chills up your spine music than somebody else's control music. The four graphs on the right show the heart rate and EMG responses from two individuals, and it's clear that the chills music increased both of these measures compared to control music, even though that same control music drove up another's heart rate and muscle tension. Test yourself. Why didn't Blood and Zatorre in 2001 use silence as a control condition? In between subjects' designs, this known uniqueness of individual brains can be a problem when making comparisons. For instance, this study was interested in motor experience through climbing and its relation to the size of the cerebellum, which is involved in fine motor control. Researchers recruited 10 world-class climbers and 10 age-matched controls with no climbing experience. Matching participants on known variables like age is commonly done in neuroimaging studies to try and minimize potential differences. Note that all the climbers were right-handed, so the matched controls were also right-handed. This seems sensible, but this practice has resulted in an extreme bias in neuroimaging research. This literature analysis by Liam Bailey and others, right here at Dalhousie University, revealed a clear participant sampling bias across several measures. On the left shows the percentage of neuroimaging studies that used adextral individuals, meaning lefties or people with no hand preference. A vast majority of the articles reviewed only used right-handed people. The middle panel with the purple bar shows the percentage of adextrals in the studies that did include them, which aligns with the 10 to 12% found in the general population. The figure on the right shows that almost half of the articles reviewed neglected to report handedness at all, so there is a lot of data still missing. When they organized articles by topic, shown in the two bars on the left, it's clear that clinical neuroimaging studies are more likely to include adextrals. However, this is still only about a third. Adextral participants were also more likely to be included in studies on children compared to adults, as shown by the two bars on the right. There are many reasons to be concerned about handedness in studies of lateralized cognitive tasks, like language and face processing. Ambidextrous and left-handed participants are a bit more likely to have reversed lateralization. But by excluding them from this research, we're not even checking to see if their handedness makes their brains respond differently. Test yourself. Why are many neuroimaging researchers biased towards using right-handed participants? Careful research design is crucial in asking questions about the living human brain. Without meaningful comparisons between experimental conditions, expensive neuroimaging and recording equipment is basically useless.